wali by yeah. Missions. Okay, good morning, good afternoon, good evening from wherever you are in the world. My name is Dr. Kanyike Francis from JCRC, and I'm your host today on this 8th gene therapy webinar series. And the big topic today is African gene therapy challenges and opportunities. But before we proceed to the gist, allow me in a special way mention that this webinar series are brought to you by JCRC. JCRC is under a big project that is EDCTP Capacity 2023. That's the umbrella project, and we have several other important partners that we are working with. We have National Drug Authority, we have Uganda National Council for Science and Technology, we have Muhimbili University of Health and Allied Sciences in Tanzania, we have University of Coimbra, Portugal. That's actually our coordinating site, and the project is funded by the European Union. We are grateful, we are thankful. So this is the 8th series, and thank you for those who have been following what we do. We started previously with introductions to gene therapy and the basics of genetics. That was Dr. Serami and Professor Jennifer. Then we went into the lab to see how gene therapy works. That was Professor Rimas. We saw innovations in gene therapy by Dr. Kevin. We had an interesting session on applications in rare diseases by Professor Almeida Lewis from Coimbra University. Then we had an interesting talk on ethics, access, and equity by Professor Karin. And just yesterday, we had testimonies in their own words. We had 2 Africans who have been cured of sickle cell disease by gene therapy. That's Jimmy and Alan. So thank you so much for those who have been following. So today the topic, like I mentioned earlier, is Africa gene therapy. What are the challenges? What are the opportunities? And our guest quantity expert today is none other than Dr. Sise Chitio. So you allow me briefly give you Dr. Sise's bio. Dr. Sise Chitio is the Executive Director of JCRC. She is a renowned vaccinologist, epidemiologist, and HIV expert with over 30 years experience pioneering ART in Africa, leading major HIV research and shaping Uganda's national HIV treatment and care strategies. Dr. Sise has conducted over 80 research projects and care and program grants for HIV patients. She was among the pioneers in prescribing ART in sub-Saharan Africa and has been at the forefront of scaling up HIV treatment in Uganda and Africa at large. Currently, Dr. Sise is the JCRC site PI for Gilead Sciences Inc. Studies and JCRC CRS lead for US NIH AIDS Clinical Trial Group, a network that has been running for more than 15 years now. Dr. Sise is keen to be involved in gene therapy. And like I mentioned earlier, she's been pioneering so many things. So it's not surprising that Dr. Sise has been keen to be involved in gene therapy as a new exciting frontier in medical research and care for HIV. So from 2020, Dr. Sise catalyzed the Alliance of Experts in Gene Therapy to bridge the existing gaps between developed and developing countries. And that instance led to the Global Gene Therapy Initiative, GGTI, that was subsequently formed to tackle the barriers to low and middle income inclusion in gene therapy development. So GGTI is working with various stakeholders to simultaneously address, identify key areas that are requisite to successful implementation of gene therapy in developing countries. Dr. Sise serves also on the advisory board for Caring Cross, a non-profit organization whose mission is to make gene and cell therapies accessible to resource-limited regions around the world. So let's all join hands from wherever we are and welcome Dr. Sise Chicho Mutuluza today to give us a presentation.
Thank you very much, Dr. Francis, for the elaborate introduction. Again, good morning, good afternoon, good evening to everybody who has spared time on a Friday afternoon in Uganda to join this webinar. So today, as already introduced, I'll talk about Africa and gene therapy, challenges and opportunities. So I'm talking about home. So traditionally, healthcare has focused on managing symptoms and slowing disease progression. But cell and gene therapy mark a fundamental shift from treatment to potential cures. So these innovative approaches target the root cause of disease at the genetic or the cellular level. Instead of lifelong medication, we are now envisioning one-time intervention that can restore normal function. So conditions such as sickle cell disease, hemophilia, and certain cancers, which were once considered lifelong burdens, can now be addressed with curative intent through gene therapy. So this transformation impacts not only patient outcomes, but also healthcare economies and delivery models. This new era compels us to rethink access, affordability, and infrastructure. It's not just about scientific breakthroughs like we have a gene therapy. It's about building systems that make these therapies available globally, including Sub-Saharan Africa and other low and middle income countries. So when we say beyond treatment, we mean moving beyond disease management towards elimination. In the next slides, we will explore what this means for Africa, the global health landscape, and how these innovations can address infectious diseases like HIV, which remains highly prevalent on this continent. Francis already mentioned the previous webinars where I'm sure they've talked about what gene therapy is, but for purposes of being on the same level for this webinar, I will just go over these again very briefly. Gene therapy is an innovative medical approach that involves modifying or replacing genes within a patient's cells to treat or prevent disease. So instead of treating symptoms like we have antiretroviral drugs, gene therapy targets the underlying genetic cause of a condition. And this can be done by adding a healthy copy of a gene, like many studies are doing for HIV, repairing a faulty gene, like many studies are doing for sickle cell disease, or even turning off or silencing a harmful gene. And again, we have gone over how gene therapy is performed, but I will very briefly repeat this. Gene therapy is performed by delivering genetic material into a patient's cells to correct or replace faulty genes. The goal is to restore normal function or introduce new capabilities to fight disease, like for HIV. There are two primary methods which we use for gene therapy, and the first one is ex vivo. Cells are removed from the patient, they are genetically modified in a controlled lab setting, and then infused back into the body. This is commonly used for blood disorders like sickle cell disease. In vivo, means inside the body. The therapy is delivered directly into the patient's body using a vector or what we would call a vehicle. Often, this is a modified virus like adenovirus or the lentivirus. And now there are many ways many vehicles that have been developed. And this carries the genetic material to the target cells. Vectors act as delivery vehicles. Viral vectors are engineered to be safe and efficient, while non-viral methods like lipid nanoparticles are emerging for certain applications. This whole process involves rigorous steps to ensure safety, precision, and durability of the genetic change that we are engineering. And this includes conditioning the patient's body, monitoring immune responses, and also long-term follow-up. Now, FDA recommends that patients who undergo gene therapy in trials should be followed up for a total of 15 years. So, and some people, even here on this call, although you have enough interest to join, may be asking, why does gene therapy matter? Gene therapy matters because it changes the way we think about disease. I think as clinicians, as doctors, we need to have a mindset shift from lifelong management to potential cures. So for individuals with genetic disorders like sickle cell disease or hemophilia, this means the possibility of a one-time treatment that eliminates the need for daily medication and repeated hospital visits. This shift reduces the burden on healthcare systems, lowers long-term costs, and improves quality of life for our patients and also their families. So in regions like Africa, where the prevalence of genetic and infectious diseases is high, gene therapy could dramatically reduce mortality and disability, but access and affordability remain critical challenges. So beyond inherited conditions, gene therapy opens new possibilities for diseases like HIV, where current treatments manage but do not cure. Imagine moving from lifelong antiretroviral therapy to a curative approach. This is why gene therapy matters. It's about giving patients a future free from disease and creating health systems that focus on cures, not just care. So we've seen in high-income countries, there has been remarkable progress in treating sickle cell disease with gene therapy. There are now 2 curative therapies that have been approved. This marks a historic milestone. For decades, sickle cell was managed with supportive care and medications like hydroxyurea, which we still use here if patients can get access, and then bone marrow transplant for those for whom a match could be found. These new therapies offer the possibility of a one-time treatment that can cure the disease. These include the CRISPR-based and the lentiviral vector-based approaches that modify the patient's own stem cells to correct the genetic defect. While this is a breakthrough for high-income countries, the question is, how do we bring these life-changing therapies to regions like Africa, where the burden of sickle cell disease is highest? Their high upfront costs remain significant challenges. Cures are being reported globally, and this gives us a spark of hope. Some are occurring naturally, and others are engineered. These cases suggest multiple pathways towards functional cures for millions of people who are living with HIV. This includes stem cell transplants, gene editing, and immune-based strategies. Each offers lessons for designing a one-time curative intervention. We will now look and review the cases where HIV cure was achieved through bone marrow transplant. And when we say bone marrow transplant, we use stem cells. We also call it cell therapy. And when we talk about gene therapy, we say cell and gene therapy. So this aspect of bone marrow transplant is the cell therapy that has led to the cures, HIV cures, of these 9 people who were living with HIV. They had a bone marrow transplant because they were also diagnosed with cancer, which was amenable to bone marrow transplant. And take off the live, whatever.
We lost Dr. Sisi.
Can you take over it? I'm sorry for that glitch, but we were on this slide for the 9 patients who were cured from HIV using bone marrow transplant, which we also call cell therapy. And these procedures replaced the patient's immune system with donor cells carrying what we call the CCR5 delta 32 mutation. And this makes cells resistant to HIV infection. So there are some people who are naturally living with this delta or this mutation, which makes them resistant to HIV infection. The conditioning, as can be shown in the in that slide, prepared the patients for the cell therapy, but it removed the existing HIV reservoir. And this included using chemotherapy and total body irradiation. So this bone marrow transplant and preparation is very intense. It's also risky. And this limits the scalability to adopt this mode of HIV cure to many more patients. But having seen these cases, and I think there are 2 more in the pipeline to make it 11, this approach has placed cell and gene therapy at the forefront of HIV cure. The 9 reported cures globally show that while rare, a cure is possible, so we should be optimistic. And they provide critical insights for developing safer, scalable, and more accessible strategies. So what would a practical one-time cure look like? We know that HIV persists in hidden reservoirs, making it extremely difficult to eradicate with a single approach. We believe that achieving a one-time cure for HIV will require a multifaceted approach, combining gene editing, immune modulation, as can be seen on that slide, and also reservoir clearance. Very importantly, all these three stages are amenable to gene therapy. Immune modulation can be delivered through vectored broadly neutralizing antibodies and CAR T cells, chimeric antigen receptor T cells therapies, while reservoir clearance may leverage the CRISPR-based platform, which is the latest tool in the box for using in gene therapy. So this goal is a scalable, safe, and affordable intervention that can be delivered in low and middle-income countries. So while gene therapy matters, we are very excited about it. We know about its possibilities. This map shows where gene therapy trials are currently happening. And the reality is very stark. Low and middle-income countries and sub-Saharan Africa in particular are completely excluded. Despite the promise of gene therapy, low and middle-income countries have no active trials. This means millions of patients in Africa remain without access to these innovations. So our push as Africa, as low and middle-income countries to become involved in gene therapy research and development must be very strong and must be collective. We can leverage lessons from scaling antiretroviral therapy and our experience with long-acting ART. It took 8 years from the time triple therapy for HIV was available to when it got to low and middle-income countries, effectively in 2004. So I would like to look at the conditions in the next slides that disproportionately affect Africa and what can we do about them as people who live in Africa. So since 1994, at least 36 clinical trials using gene therapy for HIV have been registered globally, but none of them has happened in Africa. Yet we have the highest burden, but zero participation in trials. So this is a major equity gap, and we have to address it. Nobody will address it for us. 65% of all people living with HIV are in the region, 26 out of 41 million people globally. So this concentration of disease burden underscores why Africa must be at the center of HIV cure research and gene therapy development in particular. Today, more than 10 clinical trials for gene therapy for sickle cell disease have been conducted globally. Again, none in Africa. This is another example of the innovation gap that needs urgent attention. About 8 million people live with sickle cell disease worldwide, with over 500,000 new cases annually. More than 75% of these patients are in sub-Saharan Africa. Without early diagnosis and treatment, 50 to 90% of affected children will die before the age of 5. So this, again, is unacceptable in the era of curative therapies. Since 1994, so gene therapy has been ongoing for a long time. Over 3,900 gene therapy clinical trials overall have been registered across 46 countries for many diseases, not just HIV and sickle cell disease. But again, I think there is one for hemophilia that was implemented in South Africa. So that means that Africa is completely excluded from the global gene therapy ecosystem. And despite us having some of the highest disease burden. So this gap is not just a scientific issue, it's an equity issue. And again, collectively, we must advocate for inclusion and build capacity to participate in these transformative therapies. So gene therapy, again, is not limited to rare genetic disorders. Everybody talks about rare genetic disorders. And in the US, they talk about sickle cell disease as a rare genetic disorder. It's not rare here. It's highly prevalent. So gene therapy is being developed for a wide range of conditions, including cancers, cardiovascular disease, neurological disorders, infectious diseases, as you can see on that slide. And so this breadth of application shows the versatility of gene therapy platforms and their potential to address major global health challenges. So imagine the impact if these innovations were accessible in Africa, where the burden of many of these diseases is highest. And the next two slides are just to show you the burden of disease overall. This map shows the disability adjusted life years, which is a measure of disease burden. Sub-Saharan Africa, again, carries a disproportionately large share compared to other regions. So for members who are here as scientists and innovators, how do we turn this burden into an opportunity for frugal innovation and equitable access to advanced therapies? I think the Minister for STI has always been telling us about the pathogen economy. I think this is where we can dive in because the need is very clear. The question is whether we can mobilize resources and partnerships to make gene therapy a reality for Africa. Again, this is the distribution of non-communicable diseases, which mirrors the previous slide with Sub-Saharan Africa, once again bearing a disproportionately heavy burden. So gene therapy is transforming healthcare globally, but Africa remains excluded. Our challenge and opportunity is to change that narrative. Through partnerships, technology transfer, capacity building, we can ensure that Africa is not left behind in this new era of medicine. And I will briefly touch on what I see as the challenges and what the opportunities are for us to be engaged here in Uganda, but also across Africa. So while gene therapy offers unprecedented promise, its implementation, especially in low and middle income countries, comes with significant challenges, but also with opportunities. I think where there is always a challenge, there is an opportunity. So the first issue is cost and affordability, which remain major barriers. Current therapies can cost up to 3 million. The lowest maybe is about 400,000. And this makes them inaccessible to most patients in low and middle income countries. Secondly, we have infrastructure and capacity. We need GMP facilities, trained personnel, regulatory frameworks to safely deliver these therapies. Thirdly, there is the supply chain and technology transfer, ensuring reliable access to vectors. These are the vehicles we use for gene therapy, reagents, and also manufacturing platforms. Gene therapy requires robust regulatory systems and ethical oversight to ensure patient safety and equitable access. But on the other hand, these challenges create opportunities for all of us. Regional centers of excellence can serve as hubs for manufacturing and training. Partnerships with global innovators can enable technology transfer and capacity building. And then point-of-care manufacturing models can reduce costs and improve access. So in Africa, we have a unique opportunity to leapfrog traditional models by adopting decentralized, innovative approaches, making gene therapy accessible for diseases like sickle cell disease and HIV, which are highly prevalent in our populations. So the question is not whether gene therapy will transform healthcare. The question is how quickly and equitably can we make it available to those who need it most? So I leave this question to all of you to see how you can participate. So having said all that, what are we doing? What should we be doing? What is everybody doing who is on this webinar today? How can we accelerate global access, and in particular to Africa, to these very transformative gene therapies, but also most expensive drugs in the world today. So I worked with Professor Jennifer Adair, who is at the University of Massachusetts, and we co-founded the Global Gene Therapy Initiative, which we call GGTI, as a global collaboration in 2020 to ensure equitable access to gene therapy cures for HIV and sickle cell in low and middle income countries. We focused on those two diseases because those are the ones that are most prevalent on the continent. So the mission is still to bridge the gap between innovation and accessibility for life-saving therapies. We are a partnership of over 100 members who are committed to gene therapy access in low and middle income countries. And the members span academia, nonprofit advocacy groups, industry, and also funders, including Gates Foundation. Since 2020, we meet religiously every week on a Thursday at 6 p.m. Uganda time to share ideas, coordinate efforts, and make sure that we address the gaps that exist. I invite many of you who are interested in gene therapy to reach out and join us, because we will need partnerships. We will need the whole village to raise a child or to get a cure. So the community is very central to GGTI. Community voices guide our work. They are not just participants, but decision makers. Our advisory boards include advocates, patients, journalists, lawyers, scientists from across the globe. And we have HIV and sickle cell disease advocates like Moses Supercharger, who may be known to many of you, and Evelyn Hallow, who is a strong advocate for sickle cell disease. We have global patient representatives such as Adam Castilejo, who is the London patient. This is the second patient who was cured from HIV. But very importantly, community engagement ensures trust, adoption, and also sustainability of gene therapy solutions. So again, the formation of GGTI was in 2020 to accelerate access and implementation of gene therapy as curative medicines. In the rest of the world, from 2017, gene therapy products are being approved. Right now, there are over 32. But our goal when we started GGTI was to initiate at least one phase 1 trial in low and middle income country, and this was Uganda, the JCRC, and in India. In Uganda, we have not yet, but we have made several strides. In India, they have done the phase 1 trial, but for cancer. So the GGTI approach is advocacy, research, clinical development, capacity building, training, community engagement, regulatory pathways, and sustainability. And we have come together in these meetings and looked at what are the pillars that we have to overcome, or the gaps to bring gene therapy to low and middle income countries. So success will depend on simultaneous progress in the identified 4 key areas, as can be seen on the map. These pillars are very interdependent, and none can succeed in isolation. So we have clinical readiness and implementation, which requires training healthcare professionals and building capacity. We have regulation and policy, establishing clear, efficient regulatory pathways. We have new technology development, which includes innovating cost-effective, scalable solutions, and then community outreach, which I've talked a lot about, to drive adoption and trust in the communities where we are going to implement these trials. So science must be accessible and understandable to the people it serves. Some of the initiatives we are involved in include gene therapy guide booklet for the public and the media, and this is currently being translated into local languages. But we collaborate with many people ranging from advocates, media, civil society to build the trust and the awareness, and also to be able to have these advocates explain what gene therapy is in simple language to the rest of the community. So the patients are not just beneficiaries, they are partners in shaping research priorities. So this publication, which includes from Uganda, Evelyn, and then Moses, is an example of how we amplify patient voices. And this publication is in a peer-reviewed journal. So we have brought patients together. You can see on that slide on the extreme left is the London patient, Adam. On the right at the top is Jimmy Olegare, who is one of the African patients who have been cured from sickle cell disease with gene therapy. He was the 4th patient to receive the CRISPR. And then Adam is also commonly called the London patient. So we bring these people to talk together. We have local advocates, I've already talked about them. There are many, but we mainly work with Moses and Evelyn, but there are others. And we also reach out to the community through music, storytelling, media platforms like Stigmaless Television to help fight stigma and promote education. So community-driven efforts are essential for acceptance, again, and sustainability of advanced therapies, and they should be central to everything that we do. So looking at technology, gene therapy cannot succeed without affordable diagnostics and monitoring. So some of the innovations that you can see on that slide include point-of-care hemoglobin measurement by using HB electrophoresis. This is the Hemex Health. And we already have that placed at JCRC, but also several sites in Uganda. Then we have a Ugandan who is doing his PhD with Umut in Case Western. He's also developed a very cost-effective testing platform. So I think more Ugandans engaged or more Africans to build local solutions will reduce costs, but also ensure that there is long-term viability. We are in the process as JCRC of implementing 2 gene therapy products. One is for sickle cell disease. It was developed at NIH and licensed to Caring Cross. And then a CAR-T cell product that has been developed by Caring Cross, which is in phase 1 trials in California. However, we are still looking for funding to establish like the GMP facility, the necessary infrastructure. We have some, but we don't have everything. And if we can do these things in an affordable manner, these people are available to give us technology transfer. And we are also talking to our government. Training and training and training is critical. We have to train African scientists. And one example is Dr. Lois, who has been trained and is still in training on CRISPR now, on gene therapy techniques. And here she was in the ADEA lab at Fred Hutch. Rachel is also in Fred Hutch now. And we are not able to do this if we do not have the North-South collaborations to enable knowledge transfer and sustainable local manufacturing. This team has already now developed a simplified ex vivo transduction method that cuts the costs by maybe 100 times. So again, for decentralized manufacturing, we need simplified equipment, which which makes gene therapy feasible in low and middle income countries. Professor Adair, working with Lois and working with Caring Cross, are also working on simplifying workflows for T cells besides stem cells. And our collaborator in India, during his trial for cancer, and the study was called VELCAT trial in India. Although he used a closed system, the costs for manufacturing were just about 35,000 compared to what these products cost, 400 and above. So we are also partnering and learning from both low and middle income countries, but also from the north. So the other aspect, again, about training is we've currently received a grant from NIH Fogarty to do research and training in Uganda on cell and gene therapy. We have currently recruited the first cohort of masters and PhDs. And we are again partnering with Makerere University, Alex, if he's here, and George. And we identified them because we were talking like now. So anybody who is willing to work with us, we are open to collaborate because we cannot do this alone. The other pillar is regulatory. And we are working with WHO. We have to get our act passed through Parliament, so we have to work with politicians. And I think this is in process. This is being led by the Uganda National Council of Science and Technology. So this is the roadmap. It's been a long journey. We did catalyze the implementation of this legislation, and this is now in process, and it is very critical for us to implement gene therapy in Uganda. But I think we are doing well, and we thank the National Council of Science and Technology for leading this process. Another project, which is why I'm giving this webinar today, and I think Francis mentioned it at the beginning, is called the Capacity 2023. This is a project that is funded by the European Union under EDCTP. And those are the consortium members. Again, it's trying to support the regulatory frameworks in Uganda and Tanzania, and also to increase awareness so that more people are aware. And if you're not aware about something and you get curious about it, you're not going to be engaged in this work. So this is a very useful project, and we've just gone about 18 months. These are the overall goals of that grant, and I will not go into the details. But we have regulations, we have monitoring, we have training, then we also have sensitization. Exactly what I'm doing today, sensitization. So our vision for Africa is to create a future where advanced therapies like gene and cell therapy are not limited to high-income countries alone, but are accessible to patients across the continent. This means establishing regional centers of excellence that can serve as hubs for manufacturing, clinical care, research, and training that are tailored to Africa's unique health needs. We should aim to leverage point-of-care manufacturing models to reduce costs, very importantly, and improve access, while we build local capacity through technology transfer and partnerships with global leaders. Our initial focus will be on conditions with the highest burden, and this is sickle cell disease and HIV, and also certain cancers. while we create a platform that can expand to other genetic and infectious diseases. As I already showed you, gene therapy can be applied across a whole range of diseases. So this vision requires very strong collaboration between governments, research institutions like ours, industry partners, philanthropic organizations to ensure sustainability and equity. By investing in this vision, Africa can move from being a recipient of innovation to a leader in delivering cutting-edge therapies and transforming health outcomes for millions. We are going to look briefly at the proposed roadmap for making this vision a reality. So this roadmap here outlines the steps that we need to turn our vision into a reality. And of course, more contributions here are welcome. But we need to establish GMP compliant facilities and clean rooms for point of care manufacturing, ensuring safety and quality. Training, as already mentioned, is very critical. Scientists, clinicians, technicians must be equipped to handle advanced therapies. Work with national and regional authorities to create clear and efficient regulatory pathways for gene therapy. I think this is also work in progress, at least in Uganda. We need to collaborate with global innovators because we can't do it alone. Innovators like Vertex, Caring Cross, and academic partners to share expertise and technology transfer. We need to begin with high burden diseases where we will have the most impact if we can demonstrate feasibility and also have a cure. Again, very importantly, we cannot do all this without financing, and we need to develop innovative financing strategies combining donor support. Our own government should invest, and I've been talking to them and knocking on their door, and then also industry partnerships. So this roadmap is about creating self-sustaining ecosystem for advanced therapies in Africa. This slide is part of many other slides that describe what we are planning to establish an African Center of Excellence for Gene Therapy called SGene at the JCRC. The vision is to revolutionize healthcare in Africa by enabling equitable access to gene and cell therapies through innovation, manufacturing, and global collaboration. And the mission is to advance gene and cell therapies in Africa through local innovation inclusive of training and sustainable access, empowering communities to overcome life-threatening diseases. Some of the work that we are doing towards establishing this Center of Excellence is establishing a bone marrow transplant. Here, the president was commissioning this in 2023. It's not yet operational. He also laid a stone for the gene therapy center, and we have space for it. We are waiting for funding wherever it will come from to break the ground and start construction. And this is the artistic impression of what this center would look like when we are done. So to deliver gene therapy in Africa is not something any single organization can achieve alone. So we invite all of you, all your organizations who are on this call. Every one of us can do a little bit. And when we put it together, we will get where we want to go. It requires strong partnerships across sectors, including industry, academia, our government, and the nonprofits. Again, I talk about innovator companies like Vertex. They bring cutting-edge technologies, which we are yet to develop. So we should not start from scratch when we can do technology transfer. they also bring expertise in manufacturing and regulatory pathways. I think Combria actually is starting a fully fledged gene therapy center in Portugal. And that is another collaborator that should be guiding us on what they've done, what are the best practices, so that we do not repeat the mistakes that they have made. But we are working with other institutions like King's College London, the Hore Gene Therapy Center in University of Massachusetts to provide scientific leadership, training, and research capacity. We are actually right now writing a proposal to the UK Research Institute with King's College London to work on sickle cell disease gene therapy product. But these partnerships are very important because they ensure knowledge transfer and skill development very fast. There are groups like Caring Cross, which already have products for HIV, sickle cell disease, cancer, that we can quickly work with if we have the infrastructure on the ground. We have the Global Gene Therapy Initiative, which I've talked about. These would play a critical role in addressing access barriers and shaping equitable models for low and middle income countries. National governments, the government needs to be involved. Regional health authorities are essential for policy development and regulatory frameworks and integration into health systems. So together, these collaborations will create a sustainable ecosystem for advanced therapies in Africa, so that we move from vision to reality. This slide just shows, this is the CAR T-cell product. CAR is chimeric antigen receptor T-cell product, that it's a dual CAR T-cell. It's been developed by Caring Cross. It's being evaluated in a phase 1 trial in California. Some of our staff have been there to do an observership. This is a product that they are willing to give us to implement here, but we do not have a GMP facility, so we cannot do point of care manufacturing. Otherwise, we should be ready. Our protocol has already been reviewed, preliminary review by National Council, but we need to build that infrastructure to be able to move forward. Again, I'll talk about the Africa HIV Cure Consortium Partnership for discovery, development, and rollout of HIV cure initiatives in Africa, which is headquartered in Zimbabwe. It is being funded by Gates Foundation and AIDS Funds from Netherlands. And this was formed, it is now 1 year. And these are the different objectives. Basically, this is to engage community and research advocacy coordination. And yeah, this is the same slide. So the partners in that consortium for now on the slide, and GGTI again is a partner. And we are coordinating the partner role of GGTI from here at the Joint Clinical Research Center. This is the King's College. This is the product that they have developed, non-viral and lipid nanoparticle-based delivery system for prenatal application. And they, this is now prenatal. They would give this in the liver, guided by ultrasound, between 10 to 15 weeks of gestation before stem cells migrate to the bone marrow. And alongside what they are doing, this is base editing product. We will be doing the neonates without conditioning and using antibodies to target the stem cells to the bone marrow. This is another project, again, partnership and collaboration. We are not going to do it alone. We are collaborating with a big team from Western University in Canada. We have 2 days or 3 days ago, we wrote a letter of intent to the Canadian government and there were 350 applications. Only 25 were requested to write a full proposal, and those proposals are 25 million Canadian dollars. But the consortium is really big, and we are looking at a kick and kill strategy, where the kick is a product that has been developed by a collaborator, Eric Katz, in Western University, and the kill is vectored bNAbs and also CAR NK cells that are being developed in British Columbia. So this is very exciting progress for us. Again, partnerships are key. So everything that we are doing in low and middle income countries and the problems we want to solve, like cost, simplicity, to enable gene therapy in low and middle income countries will obviously advance gene therapy for everyone everywhere. So we will continue to work to make these things easy for high income countries, including Portugal, if they have sickle cell disease or it's a rare disease over there. So I think the achievements we have made so far and the achievements we will make moving forward will be made together in partnerships, not as a single institution. I would like to acknowledge all the organizations and the people within those organizations on this slide. Some of them may not be here, but we have not gotten where we are in advocating and making some steps in gene therapy without the involvement of these people and other projects that I've mentioned, including Capacity 2023, including the African HIV Cure Consortium, and many others. And I think that's the end of my brief talk. Over to you, Francis.
Wow, wow. Thank you so much, Dr. Sise. That was an excellent presentation. I bet the audience enjoyed it because I did. I found it very informative. So at this moment, we are going to launch our evaluation form. We need the feedback. You need to tell us where to improve. Like Dr. Sise said, we are raising the child. It is the entire village raising the child. So on the other note also, I want to take your questions. Keep the chats busy. I see some questions. And Dr. Sise, if you can allow me read a few. I know more are going to come, but I can read a few and we dive in there. Marcos de Lima is thanking us for the presentation. I'm happy to read that first. People are appreciating what we are doing. So there's a question for you, Dr. Sise, from George Mwanji. He's asking, how does gene therapy ensure that only the targeted defective genes are modified without altering health DNA sequences? Also, take this from Tamale William. Do you foresee infectious diseases with standard treatment being treated with gene therapy, especially those that have drug treatment resistance? Is this going to be an alternative strategy in the management of these diseases? Then I'll take one more. You answer those as I'll read others. Kato Bridges is actually a biochemistry student. You actually answered this, but you can re-echo it again. He's wondering whether there is funding for people to take up PhD in gene therapy. So you can answer those as I read others later.
Thank you. I'll start with the last one. There are different opportunities. Right now, what we have in Uganda is through this grant from Fogarty, and we put out adverts. I don't know how you missed them, but we also were taking very few PhD candidates, and we've already interviewed and selected. However, I'm sure there are other opportunities beyond our project. And again, the project that I talked about, where we are partnering with Western University, there will be possibilities to take on PhDs, but we need to first win that one. Then we can talk about PhDs. But certainly, I think we also have many collaborating institutions. And Marcos, who made a comment there, could take a PhD student, why not? So there was a question about infectious diseases. That what? Whether we could have gene therapy for infectious diseases that already have treatment, but there is development of drug resistance. Okay. So,
Yeah, sure.
so absolutely, the possibilities are wide. It is up to us. We have to dream and think very widely. Otherwise, if the diseases are affecting us, why should we wait for other people to come and think for us? But first of all, you have to understand, you have to learn and see what gene therapy is, what can it do. But I cannot say that there is no possibility to even look at drug-resistant TB, for example, which requires even treatment for 12 months or more. Why not? So it is up to us to dream, but we dream and then we implement. We have a vision, but we have to go to reality. So we have to do something. The first question was, how do we make sure that gene therapy only focuses those genes that we are interested in and not other genes in the body? So that borders very closely on safety. If I go and develop a gene therapy product that is going to modify all your genes, all the cells in the body, that is a problem. So in the lab, and we had that session again by Professor Rimas, what is done in the lab to make sure that we only target those regions for which we want to modify or to silence. There are mechanisms of doing that, and I'm not going to go through them here on this call, but they are there. And one of the issues for safety is, are we sure we are going to target those positions? If there is a safety issue of off target, and I think there are many different strategies that people are working on to minimize off target of your gene therapy.
All right. All right. Thank you. You should be happy, George, William, and Marcos. So there's another question for you, Dr. Sise, from Brian Gisha. He's wondering, in your experience, he actually says, in your experience, doctor, what do you think is the biggest barrier preventing gene therapy from becoming a first-line treatment rather than a last resort? I see a lot of emotions in the question. There is another question from Alice. It's about finance. Alice Namale is wondering, do we have an investment case, what's in quotes, prepared for purposes of lobbying and resource mobilization, given the need? Then I also have a question for you on regulatory frameworks that you talked about. I see we have ANCAST on board. How far with bridging the gap on regulatory frameworks from other African countries? I know Uganda is already working with ANCAST. So you can take those as I read others shortly.
Okay. I think I had a slide on barriers to gene therapy. It is several factors, but maybe I can sum it all up in financing. There are several things, although regulatory is not necessarily financing. If you don't have the regulation and the regulatory framework, again, that's a barrier. So we cannot say this barrier is more important than the other. I could have my money, I could have the protocol, but I do not have the regulatory framework within my country to be able to do gene therapy. But first of all, available gene therapies, which we could adopt as a country, are so expensive. 2.5 million, 2 million, 500,000 for cancers. So we cannot, even our government cannot adopt those. How can we make the gene therapy cheaper? First of all, by innovating locally, by doing research ourselves to innovate and simplify things so the costs can come down. But how are we trained enough to be able to do that? Do we have the finances? Someone was asking about PhD opportunity. Do we have the finances to train local scientists who are working to solve a local solution? I've told you we have access to a gene therapy product for HIV. We are not able to evaluate it because we have not yet set up the infrastructure that we need. A minimum of a GMP facility, it's going to be more than $1 million US. We are not seated, we are looking, but it's not also easy for, even if you say I'm going to fundraise from local companies in Uganda, it's not easy to get that money. But we've gotten some of the things we need, we don't have everything. So if you look critically, all these things resolve about funding, but funding for what? So it's not one item that is a barrier. There are several. Regulatory may not need funding, but if it's not there, you still can't move forward. If the community, by the way, where you want to do the gene therapy, don't understand what you are doing, do you think they will accept your gene therapy? I think all the factors have to be addressed simultaneously because they all affect and they all can be very big barriers. You may think, community, I don't need money, but they don't want your gene therapy. They think you're going to kill them. They will not come. Alice, you asked about an investment case. I don't know if you remember, I showed a slide where I say African Regional Center of Excellence. We have, it is still work in progress, but we have a slide set. We have presented it to Afrexim Bank. We are reaching out to African Development Bank. We are reaching out to investors, maybe who can invest. And we are also reaching out to the inventors, especially for products that are on the market. Could they agree that patients in low and middle income countries can pay a small royalty? Even if they said yes today, and we do not have the infrastructure to do ex vivo gene therapy manufacturing, we cannot move forward. But that is a very important point. We have also made a case to the president. We have gone and presented it at cabinet, and they said they would, the cabinet agreed to fund us. We are not in the budget for this year. And you know, everybody knows what is happening, and maybe the finances are not there. But certainly, we are not going to look for funding to do this when we do not have a written document, a good document, as an investment case. And Alice, if you are available to join us, would be very grateful for any support and input from other people, because this center is for us as Ugandans, and then for the region. The other question was from Francis, regulatory frameworks. What was the question exactly? Francis?
Yes, the question was, you mentioned in your presentation that one of the challenges was actually on the regulatory framework. And I already know that here in Uganda, we are working with ANCAS, that is the Uganda National Council of Science and Technology. But I'm wondering how far with bridging the gap elsewhere in Africa. I mean, we are looking at Africa and its challenges. Do we have any, you know?
So when we first wrote to the president in 2022 to revisit this GERA Act, Gene Editing Regulatory Act, we went with a support letter from WHO. And WHO had just released guidelines, international guidelines. So WHO also supported us initially. It continues to support us. So one of the things that has to happen is that you have to be proactive about what you want. The National Council of Science and Technology, for example, will not cross to Kenya and say, have you done this? But do you know that Rwanda already has an act on gene editing? How much have they done on the ground? I'm not sure, but they already have an act. Maybe we should go and check and see what they are doing. But I think we already have, all of us adopt things from WHO guidelines, isn't it? So the Africa CDC, maybe for Africa, may need to be more proactive about engaging different countries to make sure that, again, as a continent, we are not left behind. But that's a good question. Yeah. But we are not responsible for other countries.
All right, thank you. I still have a couple of more questions. Lucy Amanyiro, actually Lucy is my classmate. I'm happy that people else were joining. So Lucy is wondering whether there's a role of pediatricians seeing the growing burden in children. Then Odul Modesto is wondering how long should validation studies take, given the high likelihood of target effects. And Benjamin Bakereha is asking whether there's any specific work ongoing in HIV and TB. I think you hinted on this, but you can echo it again. Lastly, as you take this bunch, Charles Wandera is actually happy. He's like, this is a great and a wonderful session. It's like briefly, brief mosaic in quotes about this initiative, but I also heard you talked about it. So it is a promising milestone. Thank you, Charles Wandera. So you can take those and I'll come back one more time.
Pediatricians, most of the work usually that is done with innovative things, we start with adults. Then we go to pediatrics. But pediatricians, absolutely, they play a big role, especially for sickle cell disease, gene therapy for sickle cell disease. And as I mentioned, we are right now writing a proposal to the UK Research Institute to do preclinical work. So we are not yet in the clinic, but this is preclinical work, but we are targeting in utero gene therapy for sickle cell disease, but also neonates. So the role of pediatricians would be very important. But I also talked about cell therapy, which is stem cells, which is bone marrow transplant. I think there is a big role for pediatricians, but all these 9 patients, or now they are about 11, there are some new publications that are going to come out. They are all patients that have been cured from HIV and in in Europe and the US. So if we have our bone marrow transplant unit operational, we are also lining up people who have both HIV and sickle cell disease. Can we do stem cell transplant in these patients with the aim of having a cure, a Kampala cure, at least an Africa cure for a change? So again, pediatrics, you have a big role to play. Abdul, you asked about validation studies. There is, like all of us know, a process for doing preclinical work before we get into the clinic. And preclinical work includes animal studies. And these are ways in which we validate our product before we bring it to the clinic. So this gene therapy is not going to be much longer than other products that have gone through research and development. Gene therapy, I don't know if the question was for HIV and TB. For HIV, there is a lot of work. I've already mentioned so many clinical trials that are taking place. For TB, there is that publication. Again, these slides will be available on the Capacity 23 website and also JCRC website. You can look at that publication and see what work may be performed, may be ongoing in the TB. I cannot tell you off the top of my head that this group or this group is working on TB. You know, TB is most prevalent here. So how can we use gene therapy for TB? It is you scientists on this call to think and see what we can do. For us right now, we are focusing on HIV and sickle cell disease, but that does not mean that gene therapy is limited to those two infections. The last comment was brief Mosei. No, we have talked to our president. He has requested cabinet to hear our investment case. We have presented it to cabinet. Cabinet approved it, but we are yet to see the funding. But you know, you have to keep optimistic. But we are not just waiting for our government. Our government should invest, but we are also talking to other possible funders. Anybody here who knows possible funders, please let us know.
All right. Thank you so much. I see Dr. Francis Sali is online, is our Deputy Executive Director. Actually, is adding on to what Dr. Sis said. I think this was for you, Manyiro, that gene therapy has a role in many congenital disorders. So the pediatricians should be happy. There's a role to play. So Dr. Sise, over to you again. Biaruhanga Timothy is wondering whether we are able to include computational gene therapy research into, these projects. And I also have another question. When I was reading your bio, I actually noted that you've been involved in over 80 research projects. And when you presented, you highlighted that there is a gap in research in Africa. I mean, research is being done elsewhere, but Africa, there's still a gap. So practically, how are we going to bridge this gap using your expertise, given all this research experience you have? You can throw some light there. Then there was another question from, no, you can take those and then I'll come back.
Yeah, so Timothy, you talk about computational gene therapy research, and maybe you can clarify this. Are you talking about like dry lab research that leads to gene therapy? If yes, there is now incorporation of artificial intelligence. There is incorporation of in silico work on gene therapy before you to predict if maybe the sequences that you have selected will work when you go to the bench or you go to animal studies. So I don't know if this is what you call computational gene therapy research. If it is, absolutely. It is, people are looking at it, and anybody on the call who is an expert in this area, I mean, can add. Francis, you asked the gap in research. The gap in research I'm talking about is in gene therapy. So because my work is research, that is why I am talking here today. That is why we set up the Global Gene Therapy Initiative to bring trials. We would have started trials already, but we still have to set up the right infrastructure. So I think we are utilizing our previous experiences to move forward on gene therapy. But there are many other people here who have been doing research who can join so that we are a bigger group.
Yes, thank you so much. I think this should be the last round. I see Brown is asking, scroll Brown. Brown is asking, other Chua strategies like ATIS, lock and lock. Then Justin Nachuara is wondering whether what are the most innovative engineering strategies being explored. You can be taking those as I, comprehend others.
So Brown wants to know about block and lock. What I presented was that no one, it is agreed, it is like antiretroviral drugs. We take 3 drugs, yeah, in combination for optimal treatment. I think we are also coming to the understanding that no one approach will lead to HIV cure. And when I talk about gene therapy can act as on the reservoir, it can act on binding, it can act on the immune modulation. You can use gene therapy. So the 2 combination strategies that people have been working on using different products is the kick and kill. You wake up the HIV from the reservoir, but you also have immune modulation or immune defenses to kill the HIV that has woken up and is multiplying in those cells so that you can remove the reservoir. But others are looking at a combination of block and lock. And again, the products that do that are different. You can lock your reservoir in and block it from ever waking up so that when you stop antiretroviral drugs, you will not get a rebound in the viral load. So combination therapy, even in HIV cure, and even gene therapy is the way to go. And those are some of the things that people are exploring. Justin has asked about the most recent innovation. And I hope you were there when Kevin Doxen was giving his webinar. The most advanced today is using CRISPR-Cas. You can use CRISPR-Cas as a medical scissor to cut your DNA and repair. You can use it to cut and insert. You can use it to just cut one strand of your DNA instead of both, which is called base editing. And you can also use it to do what we call prime editing. But beyond that, there are other technologies that are called epigenetics, where you are using chemicals to modify how the genes are being expressed. although this may not lead to a one-time, like, complete cure of diseases. So those are the latest methodologies today, but people are working every day. And tomorrow it might be different. So you have to read to keep up with what is happening.
All right. Thank you so much, Dr. Sise. Appears like you've answered most of the questions. I see Benjamin Bakerena is supplementing, actually saying, yes, bioinformatics is being utilized in C&J therapy. So he's been following. I think you've answered most of the questions. I don't see any more online. You've done a good job. Thank you so much. Thank you for sharing your expertise with us. It's been so engaging. It's been so informative. We are happy. So I think I'll allow you, Dr. Sise, to give your closing remarks, and then I can come back and close for today. Thank you.
Thank you, Francis. I think I gave my closing remarks in the slides, but I think the plea to everybody is that, as usual, we are being left behind in Africa. We have the biggest problems. And are we going to sit around and wait for other people to sort our problems? It is us. So we have to familiarize ourselves with the science. We have to learn what to do. We have to engage, but we cannot work in isolation. We have to work in partnerships so that we can move forward. So I'm looking forward to more of you joining DGTI, because there you learn, you listen and you learn. And yeah, thanks for attending this session. The webinar will be available if you want to go back and listen. But again, we are very open to collaboration. We are also open to funding if anybody on this call has funding.
Wow. All right. Thank you so much. It's been great. Thank you for those who've taken time to respond to the evaluation form, giving us feedback. You've done a great job. Our next webinar series is on the 24th of November, and that will be Professor Boro, sorry, and it will be a Monday, not Friday, like this has been. So allow us close for now. See you then. Let's keep the flame burning. We sign out.
Thank you, Francis.